All right, chapter 4, we are talking about carbon and the molecular diversity of life. So as you know, all living things are based around carbon molecules. So when you get to organic chemistry, junior year, you're going to be talking about all these carbon-based molecules, and these are the basis for life. So chapter 4, we're going to hit some of these big carbon molecules, molecules of life, and talk about the molecular diversity in these molecules. Okay, so we know that carbon is the backbone of life. We already talked in chapter 3 about how most cells are 70 to 95% water. Well, the rest of your cells are these carbon-based molecules. Carbon is an amazing element. It is unmatched in its ability to form these very large, very diverse, very complex molecules. And if you remember all the way back in chapter 1, we talked about those 4 molecules of life. These 4 molecules of life that we were interested in, remember, were proteins, DNA, or nucleic acids, carbohydrates, and the last one are lipids. So lipids are a little bit special in their carbon content. But if we think about 3 of those 4 biological molecules, the proteins, the nucleic acids, particularly the DNA, and the carbohydrates, these are all molecules that are carbon-based. They have these big, long carbon backbones, and they distinguish living things. They're important in living things. So let's talk about why carbon is so important to life. So one of the things that makes carbon so important is it's what we call a versatile bonder. That means it can bond covalently, so that's important. Remember, we talked about what those covalent compounds mean, what that means, and carbon bonds covalently with up to 4 other atoms. So here I've shown you this in 4 hydrogen atoms, but these constituents that we call them, that carbon is bonded to, can be anything and everything, and that's one of the things that contributes to the molecular diversity of life and how we get all these different carbon-based molecules, because these constituents to which carbon is bound can be almost anything. Carbon can also form single covalent bonds where we, remember, share 1 pair of electrons, or it can form double covalent bonds, remember, where we share 2 pairs of electrons. So again, that's something that adds to the molecular diversity that we see in living things. Okay, the other thing that makes carbon so important is that it forms these backbone type structures for these large organic molecules. So we can have simple carbon chains where we just have a straight chain of carbon, 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 carbon. And in this case, I've shown it to you all bound to hydrogens, but remember, these constituents can be anything, and that's what contributes to the molecular diversity. We can also have these branched carbon chains. So again, here we have our simple carbon chain, which is a long straight chain of carbon, but then we have a branch coming off. And now we could have another simple chain here, another carbon backbone. We could have another branch maybe coming off here, and we could have another carbon backbone. So you can see how these molecules could get very, very large. Not only do we have simple and branch chains of carbons, we can also have carbon rings. So these are really important in biology, and this line should really be like that. It shouldn't be drawn straight up and down like that, showing that this is a carbon ring. So carbon can form these ringed structures, which we also see are really important in biology and biochemical reactions. So 3 types of sort of these backbones that carbon can form. All right, so let's talk about some of the way that we represent these carbon structures. So they all have names, and I'm not expecting you to memorize this chart, but I want you to understand that there are different ways that we represent carbon molecules. So we have the molecular formula. This tells us the identity and the number of the atoms that make up the compound. So for example, for methane, the molecular formula is ch4. That means we have 1 carbon atom and 4 hydrogen atoms that make up methane. If, for example, we look at ethane, here we have 2 carbon atoms and 6 hydrogen atoms that make up methane. So that's the molecular formula. The structural formula tells us the arrangement, or shows us, rather, the arrangement of those atoms. So here we can see we have a central carbon, we have a central carbon atom bound to the 4 hydrogen constituents in methane. In ethane, we have the sort of the beginnings of a small little carbon backbone, and then we have our 6 hydrogen constituents around that. Ethylene, for example, that's a special one because we have this double bond here. So remember I told you carbon can form single covalent bonds or double covalent bonds. So ethylene has 2 carbons and 4 hydrogens, but it also has that double bond. We can also show you the ball and stick model. This shows you the geometry of the molecules. So how is it arranged? How are they arranged, and how they look in a 3D structure? So we're not going to spend too much time on those here in biology. You'll learn a lot more about those in chemistry. And then the last one is called the space filling model, and this shows you the size of those molecules and exactly how much space they take up when you take into account the size of the atoms that make up the molecules. And again, you'll learn more about those when you get to chemistry. So we've talked a lot about carbon bonding to hydrogen. Most of the images that I've shown you so far are carbon bound to hydrogens, but don't think that that's the only thing that carbon can bind to. It also binds to lots of other different partners. For example, oxygen and carbon dioxide. It binds to amide groups, these NH2 groups, to form urea. So carbon can bind to lots and lots of different substituents, and that's what makes it so versatile and so diverse. All right, so let's talk about isomers. Isomers are compounds that have the same molecular formula but different structures. So if you remember back to the previous slide, we talked about the molecular formula telling us the type and number of atoms. So that's the molecular formula. And then we have the structural formula, which showed us the arrangement. So basically, isomers are going to have the same molecular formula, but different structural formulas. And I'm going to show you some examples of these in a minute. But there are 3 types of isomers that we are interested in. We're going to talk about structural isomers. So these are isomers. So we're going to talk about structural isomers. These are isomers that have different covalent arrangements of their atoms. We're going to talk about geometric isomers. These have the same covalent arrangements but different spatial arrangements. And lastly, we're going to talk about enantiomers. These are your mirror images of each other, and I'm going to show you pictures of all of these and explain the differences as we go through. All right, so let's look at structural isomers. So these 2 molecules are structural isomers. The one on the left is pentane, the one on the right is 2-methylbutane. Both of these molecules have a molecular formula of C5H12. So if we count these, let's count them just to be sure. And we'll do our carbons in red. So we have 1, 2, 3, 4, 5 carbons, and we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens. Let's look at 2-methylbutane. We have 1, 2, 3, 4, 5 carbons, and we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens. But you can see that the arrangement of these molecules is different. In pentane, we have a straight chain carbon backbone, whereas in 2-methylbutane, we have a branched chain carbon backbone. Both of them have the same molecular formula of C5H12, but the arrangement of these atoms is different. These are what we call structural isomers. The next type of isomers we're going to talk about are geometric isomers. Now, geometric isomers, the key to geometric isomers is always going to be the double bond. So if you do not have a double bond, you do not have geometric isomers. So geometric isomers will always have that double bond. That's going to be the key. Okay, so the way that we identify geometric isomers is based around the orientation of the constituents around that double bond. So for example, if we were to take a pen and draw a line straight through that double bond, if we look at our X constituents here, the ones in X, so these guys up here, they are on the same side of that double bond. They're both on the top side of that double bond. So that is what we call, those are then what we call cis isomers, because cis means same. So they're on the same side. If we take that line and draw it down through the middle of the double bond, and our constituents are on opposite sides of the double bond, we call that trans, because trans is going to be opposite. So if you think about a transatlantic flight, you are flying across the Atlantic Ocean from one side to the other. In a trans isomers, your constituents are on opposite sides of the double bond. So remember, for geometric isomers, the key is going to be the presence of that double bond. All right, lastly, the last type of isomers that we want to talk about are the enantiomers. So these are ones that are mirror images of each other. So again, they have exactly the same molecular formula. And I should mention that the same is true for the geometric isomers. They also have exactly the same molecular formula. So again, all of these isomers have the same molecular formula, but different arrangement. So for enantiomers, we have what are called L or left-handed enantiomers and D or right-handed enantiomers. So why they call it D and not R, I don't know. I can't tell you. But they're called L isomers and D isomers. You can see we have the same basic structure. We have a carbon central atom. We have a CO2H up at the top. And then we have, you can see our other three constituents are arranged differently depending on the orientation of the molecule. So we have L isomers and D isomers. So again, this is a type of structural isomer that we refer to as enantiomer or a mirror image. Okay, so here are, this is a little chart that I want you to use when you study for your test. You will be responsible for each of the 5 groups on this test: their name, their structure, and their function. So these are constituents, or what we call functional groups, that can be attached to carbon atoms. So you can think about a carbon being bonded to each of these functional groups. And when they bond to carbon, they give that carbon molecule different properties. So for example, if we have a hydroxyl group attached to a carbon atom, that is going to make that carbon molecule polar. It's going to also be involved in linking smaller molecules to make bigger molecules. Anytime we add that hydroxyl group, we're going to induce polarity or make that molecule become polar. If we attach an aldehyde, that's going to be important in reacting with acids and alcohol. So it's going to make that molecule more reactive. If we add a carboxyl, it's basically going to turn that carbon molecule, that carbon backbone, into an acid, because remember, if you think back to chapter 3, remember what acids do. They give up hydrogen ions easily, and that's what's going to happen with these carboxyl groups. Carboxyl groups are also going to be important in the formation between amino acids used to build proteins, and we'll talk more about those in the next chapter. The amino group is actually going to turn that carbon molecule into a base. It's going to make the molecule basic, and are also going to be important in forming proteins or forming those bonds between amino acids. Lastly, we have the phosphate groups. So phosphates also give up hydrogen ions easily, so they function as an acid. And these are also going to be important in the bonds in our DNA, our RNA, and a molecule called ATP, which we will talk more about as we go through the rest of the class. So again, you are responsible for the names, the structures, and the functions of these 5 functional groups, or these 5 constituents. So you will need to know these for your test. All right, so that's the end of chapter 4. I'll see you in class.